And just an example of where physics is coming into play analyzing things. This is a GFP fluorescent focal adhesion protein. You're looking at high magnification on two focal adhesions that were bleaching a dot using fluorescent recovery after photobleaching. And we developed a technique where we can measure the unbinding constant, the K-off for this protein in vivo, in a living cell. I don't have time to go into it. And we found that if we use a drug that dissipates the tension, the pre-stress inside the skeleton, we increase the K-off by almost twofold. And you could argue that drugs do lots of things. So now we actually linked with Eric Mazur, and we used a femtosecond laser to cut, physically cut, one stress fiber and did frat on the focal adhesion at the end, and we got the exact same thing. So here's a case where actually tension on the molecules in the cell is changing the unbinding constant, okay? And physics was needed to get. Now the other side of tensegrity that people thought I was nuts, except for Don, in the beginning, was it predicted that you would have force transfer from the surface all the way to the nucleus and into the nucleus if there were stiffened elements that were linked, and forces would travel faster over a connected network than they would in solution. And I won't go into the work that we did to show that was true, but now we put a review last year. All the molecules have been identified, active filaments linked through nesprins and sun proteins to the nuclear lamina, which links to the nuclear skeleton. Intermediate filaments also link through other proteins. Link complexes are critically involved. What's important, though, is that forces really are transmitted from integrins over these pathways and concentrated and focused over these cytoskeletal pathways. Ming Wang, when he was at Harvard, we did work where we could take beads on integrins, and instead of pulling, we just rocked them at a constant frequency, and we used the phase locking technique to look at displacements of GFP-labeled proteins inside the cell. We knew the Young's modules of the cell because we could measure that, and so we could do traction force microscopy in the cytoplasm and do stress and strain mapping. And we could show that stresses were concentrated far away from the integrin, and that it was pre-stress dependent, but Ming went much farther on his own. He used a FRET reporter for the tyrosine kinase SARC, which is one of the classic chemical signaling targets for growth factors. And here you can see in the yellow and the red is activation. He's just twisting the bead back and forth here. And all the way over here and close to the nucleus, he gets activation of SARC in, I think it was 5 microseconds. Whereas if you had a chemical growth factor like EGF, it takes 5 seconds. So this is experimentally confirming that you get focusing of forces at long-distance mechanotransduction at distant parts of the cell, and that it happens faster than chemical signaling. So I had a review that was titled "Cells Act Locally but Think Globally," because the fact is that if I pull on integrins in this cell and this cell, I activate early signals identically. But this cell somehow knows that it is distorted and stretched, and it takes that signal along with many others, and it processes it and says, I'm going to grow, where this says, I'm going to die. And so we still have to understand how this signal integration works at the whole level. Now, I want to go back in vivo because people keep saying, is this relevant in vitro? And I've had that my whole life, so we go back and forth. So we went back to test this model, and one of the things in vitro did is when we mapped out how spreading regulates cell cycle progression, we came to this small GTPase rho as a critical target. It's very interesting because it does 2 major things. One is to regulate actin polymerization, the structure of the cytoskeleton. The other is ROCK, which is an enzyme, a kinase that regulates myosin light chain phosphorylation and tension. So the 2 key things in the tensegrity model. And so we then went back in vivo and used chemical modulators in the pathway. We looked at whole embryological development of a whole lung of the mouse, taken out in culture. This is the trachea, the bronchi. These are going to form alveoli. And you see there the branching morphogenesis. And if we slightly increase tension, we actually increase branching morphogenesis. And if we greatly increase tension, and we could show this is due to tension and not other factors, you literally compress the gland and it goes into apoptosis at a whole gland level. It actually dies. The other interesting thing is that, and if someone could turn the lights down briefly, this is the basement membrane in the growing normal bud. The periphery is where it grows most rapidly. This is the thinning of the basement membrane relative to the clefts that people talk about. If you inhibit tension, there's no thinning. You put the lights back on. It's just like the run in the stocking without a leg in the stocking. There's no thinning. And if you look at the morphology, this is a normal bud epithelium. There's a normal capillary sprout. If you increase tension, you get more budding and more sprouting morphogenesis. It's well known that if you pull on blood vessels, they actually grow. What's most interesting for this meeting is if you inhibit tension and you don't have the basement membrane thing, you get piling up of the cells, and they look tumor-like. You have no inhibition of growth. Same growth rates, you've lost pattern. Okay, and so I'm not calling this a tumor, but I'm saying you're getting the tissue disorganization by not having the basement membrane expand and increasing the growth rates in vivo. Now, one of my postdocs, Akiko Yamamoto, went further and she mapped out other things involved in the Rho pathway. But one interesting result was that she came out and found out that the same pathway, we work in endothelial cells and tumor angiogenesis, regulates the VEGFR2 receptor. This is the vascular endothelial growth factor, the major. This is what the major anti-cancer drug inhibits, this interaction with this receptor. She found that this pathway regulates two transcription factors, T2, TF2I, and GABA2, and they're antagonistic. And it regulates their entry into the nucleus, and this regulates VEGF2 expression. But what was most interesting is that this is mechanically controlled. This pathway we identified by cell stretching on a 2D substrate turned out to be sensitive to the elasticity of a matrix in vitro. And then we made the matrix, put it in PDMS, put it in an animal in vivo, and we saw the same thing. Optimal angiogenesis in vivo in this matrix stiffness, whereas on either sides it was lower. And we could go on and do eye development and show this was all relevant there. This came out in Nature about a year ago. So we really believe that the mechanics is critical, not only for the tumor, but also for the angiogenesis that's equally happening just outside the epithelial bud and happening just outside the growing tumor. And finally, I think you've heard mention this, but actually no one's mentioned Nina Bissell and Zena Werf's work, where they took made a transgenic mice, and all they changed was a matrix turnover enzyme, stromolysin. And those mice actually had accelerated development, and then they formed tumors. And those tumors had all the chromosome abnormalities and rearrangements that you see in classic cancer by just changing the matrix microenvironment. And then Val Weaver did work in vitro where she showed that if you could have a positive feedback loop with cells that are along the lineage of cancer, that basically, if you have a stiff matrix, the cells pull on it and get stiff and grow, and that makes them more contractile, and they realign more matrix, that makes them grow more, and it also regulates their growth factor signal, and the sort of an autocatalytic, a mechanical autocatalytic feedback loop that could lead to cellular autonomy and transformation. So we really think this is somehow involved in the process. So to end this part, I'm going to just simply say that matrix and cell cytoskeletal mechanics are key regulators of both normal and cancer tissue development. But to end, I want to move to where the specificity is. Everything I've talked about is kind of hardware. Where is the software? I think this is where physicists probably can do the most to help us out. So this is 5 or 6 years ago, a simplified view of signal transduction. It's basically a mess, right? I mean, everything connects with everything. It's one interdependent network. Everyone draws arrow map kinase, but everything affects everything else. The other thing is that if you want to switch cell phase, if you want to make a cell grow, you can't just say grow. You have to say grow, but turn off differentiation, turn off apoptosis. Same thing with everything else. So they have to be linked. And people have mapped the genomes of many, many different species. And basically, it's one connected graph over 75 to 80% of it across every species. It's all connected. So how does this work? So Sui Wang, who was a postdoc at the time, is a really brilliant guy, and basically said, we have one control parameter, we just vary shape, and we get these phase transition-like switches. They either die, or they differentiate, or they grow. And he said, this is like, water transitions in physics. He said, physicists don't talk about signal transduction. They talk about collective behavior. And so, essentially, we began to explore work, and Don Kaufman talks about this for years. It comes out of the complexity fields through Kaufman's work. Imagine you have a genome that has only 4 genes. And red is on and gray is off. So if you have 4 genes, the possibility of phenotypes at different states is 2 to 4. If you have 30,000 genes, it's 2 to the 30,000th power, which is usually like beyond anything explainable. But if you have these genes that are connected via some sort of regulatory interaction, like A inhibits B and stimulates C and D, or Stewart did with Boolean functions like not and or not if, then if you start in any state like A is on, the next time step, because A turns on C and D are on, because it inhibits B, it's already off. It's stable. If you basically start at a different point, now you can get to the same point. This is called the stable attractor state. If you go to 6 genes, it gets more interesting, and you begin to have hills and valleys like watersheds that essentially all the states will trickle towards a common point, which either could be a single attractor or a cycling attractor, such as this. The most important thing is to change a stable state from one to the other in a dynamic network like this. You have to hit multiple nodes in the network simultaneously. This is the world, but we'll come back to this. So we did a simple thing early on. We just took all the data we had accumulated from our own studies, what we knew about the cell cycle. We had 12 genes. We did Boolean network modeling, and out of it came a cell cycle. Out of it came different G0 states, whether you removed growth factor-containing medium or detached cells, or rounded cells, and that's exactly what we saw, which we got really excited about. And so we began to say that, well, rather than the current model of no link from genotype to phenotype with these different signaling pathways, and you could have the same transcription factor that does opposite things in different contexts, maybe cell phenotypes are attractor states, whether it's type lung versus liver or growth versus apoptosis versus differentiation, and then we need multiple gene switches to go across. So what we decided to do is to do experiments. This is where we move forward. To do this, we had to develop entirely new analytical tools for visualization. Everybody in biology does heat maps, and they look for what genes go up and down. We didn't really care what genes go up and down. It could be that this gene never changes, but it's critical because it has to be on this level while others go up and down, for example. So we wanted to watch how the entire genome changes over time across many different conditions. And it's very hard to look at this. So we developed our own software, which is on the web for free, called JEDI. And basically, this is a mosaic where each tile has multiple genes. It's self-organizing maps is used to generate, but each tile has the same genes in every picture you're going to see. And those genes have behaved similarly over time and experimental conditions. So for example, all genes that went up, down, up, down, up, down might be here, and the ones that were completely opposite that went down, up are here, and self-organizing maps just do the most similar. And then you could do this over time points or conditions. And now you could actually do movies, so this is from the web, a HeLa cell cycle, and you actually could see that the whole genome cycles. And interesting that at this point here it's cycling D1, which is just as an aside, kind of interesting. But it allows us to look what's going across the whole genome. So we did an experiment. We took a very well-characterized system, HL60s. These are human pro-myelocytic myelocytic precursor cells, stem-like cells. Now, it's been known for 30 years. When I first worked in the lab in 1976, on Metastasis, I should say, there are guys next to me who took these cells, and it was amazing. They came out of humans. They cultured them for years. You give them retinoic acid, they become neutrophils. But you give them this totally non-specific detergent, DMSO, and they do the same thing. And I thought, 30 years later, they figured, like, what's the critical signaling pathway that does this? Nobody's ever found it, okay? So we thought, well, what could explain it is if you basically have the same starting point and you have to hit multiple nodes in the network. Nature may have evolved retinoic acid to do that, but DMSO may hit enough things that it basically could kick it over this hilltop in a watershed idea. So the idea is that what you would see is that if this was a directed signaling pathway, you'd see liver switches going in a directed way to become neutrophil. If it's an attractor, you'd see thousands of different genes change. And then it would converge towards a common endpoint. And so, to make a long story short, this is published in Physical Review Letters a few years ago. If you use our software, or PCA analysis too, at time 0, you have the same stem cells. When you give these two drugs, they're completely opposite in 24 hours. But then they converge to exactly the same point. And this is across, in this case, 2,773 genes. And we can also do single cell analysis and confirm this is truly bistable responses, which is the other thing for the physicists. Biologists will tell you these things are going up over time. That is complete lunacy. And you know that with growth.